Makopa, I think he's supposed to be the second presenter. Yes. Yes, P2. Yes. Okay, thank you very much, Prof, for assisting. We have our presenter here is Makopa Ledimo Faith. Which one do you like? Faith, Ledimo? Okay, Ledimo is here to share with us estimation of growth curve parameters in goats, a review. Over to you, ma'am.
Good morning. My name is Ledimo Faith Makopa, and I'll be doing a presentation on estimation of growth curve parameters in goats. Growth is an economically important trait in animal production, and it's one of those subjects that can be explained with the aid of growth curves. Growth curve parameters of goats need to be estimated in order for farmers to know the right feeding program for their animal and also know the right time their animals are ready for slaughter. There are several nonlinear growth curve models that are used to predict the life weight of animals. These models include Brody, Gombes, Von Betalamfi, Jonashek, and more models. And the reason why we choose to use growth curve, nonlinear growth curve models is that the growth of an animal is in a sigmoidal shape. So nonlinear models are able to explain these parameters better than linear models. And however, there is no common nonlinear growth curve models that is used by researchers to predict the life weight of goats. Therefore, the study is gonna focus on finding the best nonlinear model for estimation of growth curve parameters in goats. And on our materials and method, we did a systematic review of data on literature that focus on growth curve models of goats, and reports that report on the growth curve of goats were used in the study, and we used Google Scholar, Science Direct, and ResearchGate to find published papers. And only papers that are between 2012 and 2022 were used in the study to make sure that the results are still relevant to the current study. And based on our results, if we go here, we will find that Gombes was found to be the best nonlinear growth curve model for estimation of growth curve parameters in goats at 28%, followed by Brody at 22%, and then logistics at 16%. Therefore, it is we can conclude that Gombes is the best nonlinear growth curve model for estimation of growth curve in goats, and it is recommended that researchers should use Gombes model when predicting the life weight of animals and growth curve parameters. This marks the end of my presentation. Thank you.
Thank you very much. It's like they practiced before they come. All of them, they don't exceed the 3 minutes. Thank you. I love goat meat. They're saying it's very healthy. Do you agree? Okay. What are the parameters that you are measuring?
When researchers are finding the growth curve parameters using these models, they are looking for weaning weight, mature weight, and turning point weight in the goats.
I thought when you're talking about parameters, you measure the egg, the head, the tail, so that is not what?
The growth curve parameters that they are using is the life weight. Like, for example, the turning point weight is the point where the animal will grow. Once it reaches that point, it means that animal is ready for slaughter. And then if you don't slaughter that animal at that point, it means the weight will decrease. Then it means you will lose your profit potential as a farmer.
Okay. Great. Oh. Okay. Thank you. Any more questions for? Okay.
Thank you, Ledimo, for an interesting study. I just want to know, besides you indicated that you considered the articles that were written between 2012 and 2022. Besides the year, what else was formed part of your inclusion-exclusion criteria?
We focused on all the studies that reviewed the growth curve models and also the studies that were predicting the growth curve parameters of goals.
Did you also include the grey literature in your search? Because I see you used Google Scholar and ResearchGate. But looking that you are maybe working on the goals, there may be maybe other reports, important reports, which may not necessarily found maybe in Google Scholar or ResearchGate.
We did the systematic review, and then we found out the papers that were used in the study were from this research databases.
I think what she meant is just to say, can you go a little bit back before technology, before ResearchGate, before?
Oh, the thing is, science keeps on developing and developing. So if you use papers that are from 19, stuff like that, you may find that they are not relevant to the current circumstance.
I won't agree with you, and your supervisor won't agree with you, but it's okay. Thank you very much. Can we give her a round of applause? Because those are your basics methods. They formed the base of what you are building on. Those are the foundations. Ne?
Yeah.
Thank you. Give her a round of applause. Thank you very much. Can we have Mona? Mona, did you present? Do we have a Mono in here? Okay, it looks like we do not have. And Rachweni? Yes, come, ma'am. Come and share with us. You guys are doing very interesting work. Rachweni is P11. Yes, P11. Here we go, Prof. Yes. Whilst they're still hanging, for the sake of our adjudicators, we have poster number P5 and the poster number P14. The students just came to us and then they requested that the posters are with the supervisor because they looked at the program and then they will be coming after lunch. So we will just have to put them somewhere after lunch. Thank you very much. We have Pretty here. You look pretty. We have Pretty who will be presenting to us bioinformatics analysis of growth differentiation factor 9 gene for egg production in poultry species. Over to you, Pretty.
Thank you, ma'am. Greetings, my name is Preeti Rachweni and I'm here to present my thesis under the topic bioinformatics analysis of growth differentiation factor 9 gene in poultry species. The growth differentiation factor 9 gene is it's an oocyte secreted factor which is essential for improving egg production since it improves the ovarian functions and the development of the egg. For this study, DNA sequences were retrieved from NCBI and they were aligned using multiple DNA sequences through Cluster W software. And furthermore, a tree was constructed using Mega 11 software. The results from table 1 show that ostrich has the lowest sequence length as compared to the other two species, chicken and turkey. And figure 1, the results of the alignment shows that chicken and ostrich aligned has a 92.3% similarity in their DNA sequences, meaning that they are highly similar in function. And results of the phylogenetic tree shows that chicken and turkey share the most recent common ancestor, given that they share the last internal node on the tree. Percentage similarities valued from 78 to 93%, meaning that they are highly similar in function and that they've evolved from divergence. This result is in line with an author who documented that the species with highly percentage similarities cannot have developed from the convergent evolution. The results of the phylogenetic tree are in line with contradicting an author who reported that chicken and turkey do not share a most recent ancestor, but this is purely because the other author used a different gene. In conclusion, the study has found out that ostrich and turkey and chicken were highly similar and that they were derived from a common ancestor, meaning that all these species can be improved similarly and they can be adapted to similar conditions. It is recommended that researchers who are interested in improving egg production in chickens might also do it similarly for ostrich, and those who are interested in improving chicken, looking at turkey, they can do it similarly. You can add the results of the phylogenetic study. Thank you.
I'm now confused, but I won't say anything. Thank you very much. That's where research is very interesting. Somebody is going to prove you wrong after 10 years, don't worry. That's research. Thank you. Do we have any question for this nice presentation? Okay, I will start here.
So if you are getting different results, is it on? If you are getting different results from different genes, can you definitively conclude that someone who is going to work with a different gene won't come out with something different from what you arrived at?
Yes, someone else can come with a different result using a different gene. But however, using this gene, they're gonna come with the same result because phylogenetic trees shows that they show how sequences are derived and not how they are similar in function. So if you were to use a different gene to look for if these animals are highly similar or if they've evolved from the same ancestor, it will be different. Thank you.
Okay, thank you, Pretty. I'm not sure as to whether I do understand you, my sister. The similarities of the three, you said they were found that they are from the same ancestor. Do you want to say the same family, like as we call them a fowl, F-O-W-L, or are you talking about ancestral as you are talking about it? You want to say the same family or what? Just to clarify me there.
Okay, thank you for the question. By the way, the similarities do not define the ancestors. There are two different statistical analyses. The first, the alignment shows that they are highly similar if they have highest percentage similarities. The second study, which is the phylogenetic study, which is the tree, shows how they are derived from each other. So it shows that the chicken and the turkey, they share a most recent ancestor as compared to that of the ostrich. Thank you. Any other question?
It doesn't look like that. Thank you very much, Pretty. Thank you very much. That's where research is very interesting. Eh? You don't tweak the results, present them as they are. Let's go to our program. I think we do have the last poster, P13. 13. Yes, we have got 3 presenters there. Yes, Sekiti. Can we have the poster? Sekiti, Libilo, and Tekwani.
Good morning. My name is Sigidi Pelkoja. I'm with Makoma Faith Tekwani. We had to present our research based on estimating soil organic carbon using laboratory-based spectroscopy at Cykerfield Experimental Farm. Soil organic carbon is a carbon that is found in the soil organic matter, of which contributes into the carbon sink and also can contribute into the soil fertility. Our study was done at Cykerfield Experimental Farm, and it aims to investigate the potential of laboratory-based spectroscopy. The first objective is to identify the key wavelength that is used to estimate the soil organic carbon. However, the second one compared the performances of 4 different spectral transformation, which is the raw spectra, first derivative, second derivative, and also the log transformation. We collected 74 soil samples and used 30% for validation and 70% for calibration, which were taken to spectroscopy lab for analysis. We used palettes to investigate our results and analyze the different transformations. I'll give to Makoma to discuss the results.
Thank you. For the results, the first diagram shows the wavelength of which the peak maxima that the VIP threshold we used were chosen as the most important wavelengths, whereas the second diagram shows the overall comparison of different spectral transformations, wherein the first derivative gave the best results. To conclude our presentation, the study has shown that the visible near-infrared and the middle infrared can be used to estimate SOC. Also, the study has portrayed that the spectral transformations, raw, log, first and second derivative can be used to estimate soil organic carbon using partial least square regression as models. And also, from what I have said, the first derivative data transformation produced the best performing SOC prediction based on its R-square, RMSE, and the RPD. And overall, the findings revealed to us that the laboratory-based spectroscopy has proved to be a rapid and a robust in estimating soil organic carbon content. Thank you.
Thank you very much. It's who and who? Because you're supposed to be 3. So it's Sikiti and Tegwani. Okay, thank you very much for your presentation. Do we have any questions for the presenters? Oh, I see. Okay, let's start here.
Hello. Thanks for our students to present their very critical, very important, study in terms of, checking the. I'm very much interested in the, lab vis-a-vis the farmers who always collect their soil for doing soil analysis. So in this, how can you recommend for how this study can help a mere farmer who is in a very rural-based, community? He wants to start farming, and he wants to start first by analyzing his soil. What can you recommend from your study? Thanks very much.
The study critical focuses on how soil organic carbon contribute to the soil, of which a rural farmer can use this study to know that the soil is fertile and healthy for his or her own crops.
Yeah, no, without taking a time, just for interest sake, you know, I'm looking forward for this typical kind of studies where, you know, we have a lot of, you know, many people are more interested in getting into farming. And then most of the soil, you know, are not so, you know, fertile for, you know, crop productions. So normally we encourage farmers to start to analyze their soil first. But one of the key problems or challenges is to get an effective, you know, instruments that can able to, you know, help the farmers at the rural base. So, yeah, but I know, you know, it's very critical that this is a lab, you know, study, but I was just checking on how this farmer can access that. Thanks very much.
Okay, if the farmer is interested in analyzing the soil in their farms, they can go look for a spectroscopy where they use the reflectance of the soil to estimate the soil organic carbon and other organic matters that are present in the soil for the fertility and other matters that contribute to the best crop production.
Thank you so much for your study. My question is just on the accuracies of those four models. You said the first derivative is the one that is performing better. What was the actual accuracy of that? And also, in the wavelengths, which wavelengths were the most important for that first derivative? What, is it visible? Is it near infrared, or is it the shortwave infrared? Thank you.
Okay, thank you for that question. Wait, can you repeat your question? So can you repeat your question?
So first question is your four models, your raw spectra, log transformed, first derivative and second derivative. Then you said first derivative is the one that performed better, right? What was the actual accuracy there? Can you read it? And also, is it acceptable? And then number two, in the wavelengths of that first derivative, which region was the most important if you look at the spectra from 400 to 2.5? Thank you.
Okay, looking at the first derivative, the most important wavelength was ranging from 400 to 2332. It was between 1040 to 1120 nanometers and 1751 to 2042 nanometers. And also the electromagnetic spectrum, the electromagnetic spectral band that gave the most accurate results on the first derivative was the near infrared. Thank you.
Okay, thank you very much. We can do the follow-up when we are eating lunch. Okay, thank you. Thank you very much to the presenters. We have the last presenter, Makuchisha. Do we have the students? It's not here, ne? Okay. Then this marks the end of our poster presentations, unless someone is here with a poster and she or he didn't present. You can come forward. Otherwise, this marks the last, the end of our poster presentations. And who is that? Who's that? Matlala. And which poster number is this? That will help us. Yeah. So we have P4, it's Matlala. It's going to hang the poster.
All right, thank you. Good day, all. The title of our research is the assessment of wastewater reuse in the Mankweng area of the Polokwane local municipality. So a quick background, we look at the water scarcity and climate change and pollution, which exacerbates water supply and hinder sustainable development. This because of social and economic development, which attracted population influx and growth that led to the increase of water use in the area. So wastewater reuse and and others are being explored, to minimize the water shortages, which is deemed as one of the feasible sustainable water resources management. The aim of our research was to assess the wastewater reuse perceptions in the Mankweng area. Objectives was to investigate perceptions of the community in the Mankweng about the recycling of treated wastewater, and also to explore this for non-potable utilization in the area. Our research is research design included both qualitative and quantitative researches. A sample size from household estimation of 10.03 from the census 2011, and so from a 95% confidence interval, and a margin error of 0.076. The sample size of 164 was used in the study because the household arranged in a grid plan pattern, systematic random sampling was utilized. Questionnaires were distributed, collected, data captured, and analyzed using descriptive statistics. From the results, we see that there are more females to male respondents, and the percentage of unemployment status shows that there are more people who are employed in the area. The table 1 shows the percentage of those who agree on the reuse of non-potable uses, but these people disagree when coming to options of using this water for cooking, drinking, and swimming. In conclusion, households in Mankweng are aware of and are experiencing water shortages. They have been implementing household-level adaptive responses, which enabled them to reduce the pressure on accessible potable water. The study highlighted the role of wastewater reuse in the municipality, and this alternative uses of wastewater is expected to elevate pressure on the wastewater treatment plants in the province. Thank you.
Thank you, Mr. Matlala. Thank you. Do we have any questions for Mr. Matlala? Treated water. Okay, doesn't look like we do. Okay, you can give him a round of applause. And thank you very much. Do we still have anyone who was left behind? Yeah, I think we are now done. I will have to talk to Prof. Mandiwana before I can say anything, or Prof. Masuku. Okay, thank you very much. I'm told that we can use the projector. The lunch is supposed to be at 1 o'clock. So what is going to happen, I'm going to hand over to the chairperson of the first session, who's Dr. Hadebe. Then he will continue with the PowerPoint presentations. Thank you so much.
Okay, it is now afternoon, and I think we can squeeze in maybe 2 or so presentations before 1, because presentations are they are 10 minutes each with 5 minutes questions. We are going to switch off the projector that's here and then use the original projector that we had. It might take time, meaning that if that takes a lot of time, maybe we're going to squeeze in 1 presentation. And that presentation is going to be from our first speaker. I think I'll need to just double check. It was Nepali, so please be ready as much as possible. I'm going to call you. For now, I'll just take a short break and sort things out before I call you. Thanks. We'll use this one for this. Yeah. Okay. Okay, my bad. I've been reliably told that we are going to use this one for now to grab 2 presenters. Can I have, please have Ms. Nepali come up to the stage, please? Okay, so I'm told this is for. Can you please get a pointer? This one is not working. We need an actual pointer.
Space for USB. Most ID.
What? You open the slide.
Open the slides.
Okay, okay.
Should I put it on slideshow?
Yeah, let's do.
Okay.
If you see me standing, you've got 2 minutes. If you see me standing for the last time, you've got 15 seconds, and I'm coming to grab you.
All right.
Thanks.
Good afternoon. Okay. I'm Nepali LE.
I'm here to share with you a study on evaluation of heavy metal contamination in sediments and risk assessment of 2 urban rivers, Limpopo Province, South Africa. I am a PhD candidate at the Aquaculture Research Unit under the supervision of Professor Moyo and Professor Rapaza. Let me take you through the introduction of the study. Heavy metal contamination is a major concern in aquatic ecosystems globally. They are normally present in trace amounts, mainly due to elevated anthropogenic activities such as agricultural runoff, mining effluent, and sewage effluent. Heavy metal contamination is common in urban aquatic ecosystems. This is mainly due to rapid urbanization and industrialization. However, there is scarcity of information in developing countries regarding their level and impact of heavy metal contamination. Sediment act as a sink and a repository for heavy metals. The suspended heavy metals in the sediments tend to be, the heavy metals in the sediment, they get resuspended into the water column. And the resuspended heavy metals in the water column, they become toxic to aquatic flora and fauna, like the plants, the reeds, macroinvertebrates, and fish species. Heavy metal contamination has been reported to be higher in sediment than in water column. It is therefore prudent to determine the contamination in sediment than in water. However, most studies undertaken in Southern Africa, they focused in contamination in water. Coming to the materials and method, the study was conducted at the Sand River and the Blood River that are in the Polokwane city in Limpopo Province, South Africa. The Sand River flows at the edge of the Polokwane city, while the Blood River flows at the edge of the Sishiu township. Both these rivers, they are recipients of a poorly treated sewage effluent from the Polokwane wastewater treatment plant and the Sishiu wastewater treatment plant. In this study, reference sites were selected upstream of these treatment plants. The Sand River has been reported highly polluted, mainly from organic pollution. And these are the studies that have conducted during the rainy season, which is from January to April, and the dry season, which is from May to August. The samples were kept in a freezer during the analysis, during transportation from the sampling sites to the laboratory. And the digested heavy metal sediments, they were analyzed using an inductively coupled plasma optical emission spectrophotometry. And all the chemicals that were used were analytical reagent grade. To assess the heavy metal contamination in these 2 rivers, different indices were applied, of which the first one is the Geo Accumulation Index that was calculated using this formula. The Geo Accumulation Index is classified into 7 grades, meaning that values below 0, they are regarded as uncontaminated, whereas values between 3 and 4 as heavily contaminated. The enrichment factor also was used. This was used to determine the extent of heavy metal contamination compared to the background area. And in this case, iron was used as a reference material. The enrichment factor is also categorized or classified into 7 groups, where values below 1 are regarded as no enrichment. With the contamination factor, this was used to determine the level of contamination in the sediment of these 2 rivers in relation to the control, of which in this case, the control are the sites that were selected upstream of the point of discharge. And this is the formula that was used. The enrichment factor, the contamination factor is categorized into 4 groups, where values below 1 are regarded as low contamination. The study further assessed the potential ecological risk of these rivers, where the risk index and the potential ecological risk factor were calculated. Both the potential ecological risk factor and the risk index, they are also categorized into different groups, where the risk index values below 150 are regarded as low ecological risk, whereas the potential ecological risk factor below 40 are regarded as low ecological risk. The study further analyzed the cluster analysis and the person matrix. This was done to group the sites with the similar source of heavy metals, and also to identify the metals that are related and also that are emanating from the same source. This was done using an SPSS. Coming to the results, the heavy metal concentration in the sediment, they followed this order where iron manganese dominated the concentration. This is mainly due to the geological formation in the catchment area. And other metals showed spatial variation, whereas others did not show spatial variation. There was no seasonal variation. The cadmium, chromium, copper, lead, and zinc, they were below the probable effects level, according to the Canadian Council of the Ministers of the Environment. This is another challenge in South Africa, that we do not have our own sediment guidelines, so we tend to depend on other countries, in this case, which was Canada. However, the CCME, it doesn't have all the PEL for all the metals. When it comes to the Geo Accumulation Index, all the assessed metals, oh my God. All the assessed metals show the Geo Accumulation Index values that are below. Can I please be assisted? Okay.
Can you get some assistance with the projector? I would say take a seat. It might take long. You can take a seat next to me here somewhere. And rest. Time-wise, I've sorted it out. Guys, sorry, we've just had a problem with the projector. So this could probably be the only presentation that we're having in this session, looking at what's happening right now. Thank you for your patience.
After I heard that we go for lunch, so that they start connecting these ones, then